It's my turn for the final presentation. I'm going to present on the biotechnology of growth promoting microorganisms for an improved plant health in vegetables. And here, if you see,
Adam,
there are,
we cannot see
hello.
your presentation.
Oh, you're not seeing? Okay, I'm sorry about it. I thought, thanks, Davide.
You're welcome. Yeah, now it's okay.
Okay, great. So here in this case, whatever the molecular techniques that we apply for discovering or else for identifying a growth promoting microorganisms could also be considered as a biotechnological aspects. So in that regard, I have actually given this title as Biotechnology of Growth Promoting Microorganisms, and how these growth promoting microorganisms can improve plant health. And in our previous presentations from Davide and Professor Muthraj and Ravindran, and from Dr. Shankar, we have seen that how the soil health as well as the range of techniques available for improving plant health. And here I'm going to talk specifically about some of the organisms that has been identified as growth promoting microorganisms and how that could have a potential to be used as a biofertilizer and as well as the biocontrol agents. Here, if you see, in case of plant microbe interactions, it's a though we have diverse microorganisms in the rhizosphere region, but still we may have a prominent microorganisms which may have either positive or negative interaction with the plant. And here, if you see, in case of microorganisms, when they have a pathogenic impact or when they have a negative impact on a plant, then we know that it is a disease and which could be caused by a fungi or bacteria or by a virus. And here, that kind of interaction is known as antagonistic interaction. And at the same time, the plant and microbes, they also have a mutualistic interaction wherein the microorganisms, they will be benefited from the plants by taking the nutrients such as carbon sources, and in turn these microorganisms can also benefit or can also support the health of the plants by various mechanisms. And in this case, if you see the beneficial microorganisms that has mutualistic interaction with plants or endophytes and plant growth promoting rhizobacteria and nitrogen fixers and phosphate solubilizing organisms. And between this antagonism and mutualism, there is also competition and neutralism. And the between the plants and microbes, there will be a competition as well for the nutrients, but however, that will not have any pathogenic or negative impact on the plants. But at the same time, in case of neutralism, the microorganisms and plants, they will also not have any interaction in the sense they will not affect the growth of the other. So here, what we are interested in this interaction, the beneficial interaction or mutualistic interaction. And here in this case, as I told you, there are beneficial microorganisms, mostly the bacterial groups of microorganisms, and there are some fungal microorganisms such as mycorrhizae, and in that mycorrhizae group we have ectomycorrhizae and the endomycorrhizae. And apart from that, we also have some of the fungi such as trichoderma and the other non-pathogenic fusarium microorganisms, which also known to have some sort of benefit to the plant health. So in that context, if we see what is plant growth promoting microorganisms and the microorganisms that are associated with plants and exert beneficial effects on plant development, and those are referred as plant growth promoting microorganisms. And here, in this plant growth promoting microorganisms, though there are wide diversity of the microorganisms, some of them could be cultured and some of them may not be able to culture, as indicated by Shankar. So in this case, in my presentation, I'm going to more concentrate on the bacteria, that is plant growth promoting rhizobacteria and how that could be used for improving plant health in vegetables. And here, if you see the plant growth promoting rhizobacteria, this is the bacterial community which predominantly present in the rhizosphere and promote plant growth. And most of the plant growth promoting rhizobacteria is used as biofertilizer and biopesticides. And we would have also seen some of them in the market. And here, these microorganisms or these plant growth promoting rhizobacteria is isolated based on a specific medium. And then we may have to identify the elite strain or else the best one as explained in case of the plant breeding by Ravindran. In that case, if you see in case of plant breeding, you have to identify the elite germplasm, which could offer disease resistance or pest resistance or abiotic resistance. And here also, we have to identify these plant growth promoting rhizobacteria by use of different molecular techniques, as well as identifying the genes that are the structural genes or functional genes that has been already known to us. And in case of endophytes, and these endophytes, they also exist within the plants. And here, these sort of endophytes naturally evolved, and it would have entered through the root system. And some of them could present on phyllosphere or on the leaves, and some microorganisms have also been noted on fruits or on the coposphere. And here, these endophytes or microbes, they reside inside plants without harming plant growth and development, but associated with plant health under normal and challenging conditions. So most of these endophytes and the plant growth promoting rhizobacteria, and most of them could be bacteria, but still there are some fungal, and that has been reported from tree types, which has a lot of potential to improve plant health as well. In case of plant growth promoting microorganisms or plant growth promoting rhizobacteria, if you see, there are umpteen number of microorganisms have been reported for its plant health properties. For example, Acetobacter, which is known to have the nitrogen fixation, then Azospirillum, and this is a very good bacteria for phosphorus solubilization, then Bacillus, and in case of Bacillus, there are so many species of Bacillus, and which has been used over the years to develop as a biopesticide or as a biofertilizer, and Panibacillus, and of course Pseudomonas. And in case of Pseudomonas also, there are so many species like Pseudomonas fluorescens and Pseudomonas chlorophyllus and Rhizobium. And in case of Rhizobium, we all know that the Rhizobium, it has mutualistic interaction with the legumes, wherein it fixes the atmospheric nitrogen and also streptomyces. And here, all these bacterium species here in this case, with respect to plant health, we are specifically looking for those microorganisms, or else we could use a common medium to isolate the microorganisms that are colonized in the root region of a plant, so that after isolation of those microorganisms, we could use the biotechnological or the molecular techniques to screen individual microorganisms, like how we are going to screen the different germplasms for selecting the elite varieties. And here also we could use the molecular techniques to select the elite strains. In case of fungus, as I told you, the mycorrhiza and as well as the trichoderma have been regarded and studied as plant growth promoting microorganisms. And here, what we have done, we would have isolated here in the island by collecting soil samples from forests as well as from different agricultural ecosystems. And we have isolated more than hundreds of bacterial strains from the rhizosphere soil sample. And of that, we try to screen those microorganisms against vegetable pathogens, and we got some interesting results, and that's what I'm going to share with you in this talk. Here, most of these plant growth promoting microorganisms have been studied as biocontrol agents in plant disease management. And here, If you see on the right-hand side, and that is one of the bacteria that we have isolated from the rhizosphere. And we have different pathogens, tropical pathogens that are affecting range of vegetable crops, starting from tomato, hot pepper, and cowpea, and sweet pepper. And of course, we have a banana in the region. So here in this case, here we have Phytophthora capsaicin in this slide. And this Phytophthora capsaicin is a very serious pathogen affecting hot pepper cultivation as well as sweet pepper cultivation in the region. And being a soil-borne pathogen, and this could devastate the entire crop, and it could cause harm at any stage of the plant. So that's why this is a very serious pathogen, and we are working on this pathogen. And here, if you see, when we tried to screen the number of strains against this Phytophthora capsaicin, and we have seen that this has shown a very good effect against this Phytophthora capsaicin. And not only that, we have also screened against several vegetable pathogens that I will present it to you during the later part of my presentation. And here, these plant growth promoting microorganisms, and they exhibit some sort of antagonistic activity and some sort of competition against these pathogens in preventing the growth, or those could arrest the growth of these pathogens. Now, how do you identify the genus of a plant growth promoting microorganism? I know that we have targeted and non-targeted methods, but here, if you see, in case of conventional methods, as Davide explained, we have phenotypic methods or colony-based identification methods are based on grow in specific media. We can have several medias, and using a specific media, we could grow that microorganisms. And also, there are microorganisms which are sensitive to the antibiotics, and based on those biochemical markers also, we will be able to identify the plant growth promoting microorganisms. And this is one of the conventional methods. And based on the microscopic techniques, we'll be able to, based on the shape or based on the size, we'll be able to narrow down the identification process. And there are some biochemical tests also available, and which has been very well explained in some of the textbooks. But at the same time, if you see, those techniques have been used several years, but as said in the previous presentation, it requires some sort of expertise or the experience to identify or discover the organisms and what could be the genus. And even if that is the case, that always resulted in uncertainties and difficulties with the identification. So in this regard, if you see, or if you ask me, what are the molecular techniques available to identify the genus of a plant growth promoting microorganism? And here, the molecular techniques and advances in sequencing types have really enabled the appropriate identification of this plant, either plant growth promoting rhizobacteria or an endophyte, or to study its genetic diversity. And here, similar to the isolation of plant growth promoting rhizobacteria, and there is a protocol for isolation of endophytes from plants as well. And once we have isolated, and once we have screened them in lab against a number of pathogens, and once we see that, okay, this particular microorganism or this strain is giving very good control against pathogen or against a number of pathogens, then the next approach, as I told, the identification. And here in this case, molecular methods are highly useful in identification of the microorganisms. And in this regard, mostly they are identified based on ribosomal RNA gene amplification. And here we are specifically targeting, and after amplification, after sequence, then it has to be blast. And based on the database, then we will be able to confirm, or else we will be able to identify the genus of the organism. And in some cases, what has been done, and as explained, sequence characterized amplified region by Davide, and we could have the ITS region, and based on that ITS, or based on that ribosomal RNA region, and the primers would have been developed for the specific identification of a genus, maybe a bacillus or maybe a pseudomonas or maybe a rhizobium or whatever organism it is. And in that case, we could use that genus or gene-specific amplification. In some instances, there may be some genes which are very specific or unique to the microorganism. In such instances, we will be able to amplify that gene, and based on that, we can say this is going to be this genus, or this would be this genus. Then now, because of the wide diversity of microorganisms, even in molecular methods also using this ribosomal RNA genes, though it has the variations, as well as using the ITS, though it also has the variations, but still we may not be able to identify them at subspecies level. So when we are not able to identify them at subspecies level, then we can also think of going for the multi-locus sequencing type. And using this multi-locus sequencing type, we can sequence the different segments of the genome, and based on that, we will be able to identify them as the genus at the subspecies level. And here, mostly the housekeeping genes are used in identifying the organism at subspecies level. So the most common target for identification of a fungi or bacteria, as I told you, and as told in the previous presentations, it is ribosomal RNA. And of course, all organisms contain these loci, and they have very highly conserved in sequence, as well as divergent enough to differentiate between the genus and species level. And here, if you see in case of prokaryotes and eukaryotes, and they synthesize and they make ribosomes. And here, for your understanding, I have given the RNA genes that are present in prokaryotes as well as in eukaryotes. And here, in case of prokaryotes, if you see the bacteria, it has 16S rRNA, that is known as small subunit RNA. And here, in case of eukaryotes, we have 18S and 28S. And here, in case of eukaryotes, in between this large subunit and small subunit, we have ITS region. And based on sequencing or amplifying and sequencing of these ITS regions, we will be able to identify what could be the genus or what could be the species. And similarly here, in case of 16S or RNA, by amplifying the whole gene, or else there are some specific variable regions. There are about 9 variable regions, and of these 9 variable regions, that lies in the whole genome or gene of that 16S or RNA, and we can amplify the whole gene, or else the variable regions can also be used for the identification of a particular organism. So here, there are different universal primers have been designed and is available for use to do PCR, as well as then send it for sequencing. Then after you get the sequence data, then you have to do it with the blast to identify what is the organism. And as I told you, here you have this whole region, that is the 16S or RNA gene from a bacterial genome. And here, if you see this gray shade boxes indicates they are the variable regions, and in between the variable regions, and you have the other regions. And here, if you see this 27F and 1492 R, so that is actually based on the location of the bases, and based on that, these primers have been designed. And in between, you also have this V3 and V4 and V5. And here, for in case if you are going to study the diversity of the microorganisms as explained by Shankar, you may need to do the specific regions. For instance, some of the regions, they can cover the microbial diversity or bacterial coverage up to 60% or 70%. But for the genome identification of a particular bacteria, I would suggest you have to do the whole genome, the whole gene sequencing of 16S or RNA gene. So once you do that, then you will get the sequence data, and that sequence data can be blasted in BLAST, and you will be able to to identify what is that bacteria or what is that organism. And here, just for your reference, I have given the primer sequence which you could use to amplify the organisms of interest from your region. And also here, suppose if it is fungi or yeast, then we cannot use the 16S rRNA that has been the same primers that has been used for the bacteria. Then we have to look for the different thing. And here, mostly the ITS primers are used for the identification of fungi or yeast strains. The reason for saying yeast strains, in addition to fungi or bacteria, the yeast strains, they also used in post-harvest biocontrol, and it has been intensively studied in some parts of the world. And so in that regard, we could use ITS primers. And these ITS primers, as I just explained before, it lies in between the large subunit and the small subunit of rRNA. And here again, they have identified the regions, and in that they have the primers like ITS1 and ITS2 and ITS3 and 4 and 5, and you could use ITS1 as the forward primer and ITS4 as the reverse primer, so that you can amplify that region. Then again, when you get the sequencing, as we all know that the sequencing cost has come down, and it for sequencing this ITS or the 16S or RNA gene for a sample of a particular amplified fragment, it just costs about $8 or $10 US. So it's not much. So in that regard, we will be able to sequence these specifically isolated microorganisms from the rhizosphere region, and we could identify those microorganisms. Also, for your reference, I have given the primer sequences which could be used for amplification of a DNA fragment, and that could be sequenced, and it has to be blasted. And here, of course, we all know that, but since there will be a different level of participants in the seminar, in this webinar, I thought it would also be good to just show that in case of blast search, of course, that is a NCBI database, and there are so many databases, but mostly I use this NCBI database for the other microbial diversity studies, as well as the metagenomics or other things. You may have a specific tool and the annotation tools, as well as the alignment tools. So probably you may use that mapping, and so many tools are available. And of course, we should also have some bioinformatics knowledge when we go for this downstream analysis of the microbial diversity. But in this case, we just need only a basic understanding of some of these databases. And here you could go to this database, and you could do this blast, and there are so many options available. You could blast the nucleotide data, as well as you could blast the protein databases as well. And also, if you have a specific gene, then you could use that DNA sequence information to deduce the amino acid sequence, and so that you will be able to identify the function of that encoded protein. So, but that may not be possible here in identification or in while you are using 16S or RNA genes. But suppose if you are using some other genes to identify the microorganisms, as explained in case of Davide, because in that case, for instance, in identification of some of the fungal strains, you may also use, in addition to ITS and cytochrome oxidase gene. So that is also used in identification of a fungal microorganisms. So in such cases, if you are using this cytochrome oxidase gene or other genes, then you can use that DNA sequence to translate into amino acid sequence. Then that could be blasted in this database, and you could find what is the function and what is the homology of that gene and the organism. And here in this particular case, since we are interested in simple identification of a microorganism. Then once you do this nucleotide base, and you have to feed the either 1000 and 1500 base pair of the DNA sequence, then you will get the search in the database. Then finally you will see what is the organism that you're trying to study, or you're trying to identify. And here, if you see, the data is showing in 99% homology or 100% homology, because this is the data that we have already submitted in this database. That's why it's showing 100% homology. And this is showing it as a Bacillus subtilis strain. So based on that, you could know now, okay, the strain that you're, or the organism that you're going to work here is a Bacillus and subtilis, or Bacillus amylolic or some other strain that you're going to work with. And here, as I told you, in addition to the 16S rRNA genes, and there may be some genus-specific rRNA intervening sequence-based primers would have been developed for a specific genus. In this case, you may find the primers for Bacillus or Pseudomonas, or many of the biocontrol strains, or many of the plant growth promoting microorganisms. And if you, based on your initial identification, if you have known that this is a Bacillus or Pseudomonas or Acetobacter or some other bacteria, then you could use that specific genus to identify that. And here what we have done is we have used the Bacillus specific primers, and while we use that, and we amplified 546 base pair of the strains, and out of 100 rhizobacteria strains that we have isolated, and we found that 47 strains belongs to Bacillus species. And similarly here, I have given the primers that could be used for the amplification of Pseudomonas fluorescens specific species. And when we did this, and because we would have given the strain name, and when we have done the sequencing and after the blast, and we found that they belongs to different species, and here ranging from Bacillus cereus and Megatherium and amylolic based on its homology. This is an example how we could use the 16S rRNA intervening sequence specific primers to identify the organisms. And as I told you, still there are some concerns with the use of this 16S rRNA primers to identify the organism at subspecies level. So in that case, what we could do is, as I told you before, we could use this multi-locus sequencing type, and this will enable us to identify the microorganisms at subspecies level. In case of Bacillus, if you see, there are Bacillus subtilis, and after that there are so many subspecies level, and based on its strain distinction and unique properties, it has been identified and it has been grouped. Here, in this multi-locus sequencing types, for instance, here there is a gyrase gene and purine synthesis gene. So there are so many housekeeping genes, and by amplifying and sequencing those genes, and there are databases, and using those databases, we will be able to identify where our strain fit into, or where our organism is fitting into, and so that we will be able to identify at subspecies level. Now, we have identified the microorganism. Okay, we know that this is a particular microorganism. It could be Rhizobium or Bacillus or Acetobacter or whatever it is. Now, as I told you, in case of screening of the plant growth promoting microorganisms, assuming that the microorganisms that we have isolated from the rhizosphere may have some growth promoting activities or may have some antibiotic activities, we have tested it against the pathogens, and we found that some strains are showing the greater activity, and those strains have been sequenced and identified as the same group of microorganisms. And if you see the general mechanism of plant growth promoting microorganisms in improving plant health, one is the growth promotion. And in case of growth promotion, it could produce the phytohormones, and it could also mitigate the stress imposed by biotic and the abiotic methods, or abiotic stress. In case of antagonism, of course, the growth promoting microorganisms, they are known to produce the antibiotics, and those antibiotics are synthesized either ribosomal genes, or they are synthesized through non-ribosomal peptides. Such as lipopeptides, and also they produce a lot of secondary metabolites, and those secondary metabolites may have suppression on the growth of the pathogens. Then the third well-studied mechanism of this growth promoting microorganisms is the production of lytic enzymes such as chitinases and beta-1,3 glucanases. And here what happens, this chitinase and glucanases that can act on the cell wall or the fungal pathogens, and that could actually suppress the growth of the pathogens. Then the final thing is induction of defense enzymes or responses in plant diseases against in plants against diseases. So based on these four mechanisms, we can look for the strains, whatever we have, and we can look for whether those strains, they exhibit any of these mechanisms. The reason is we cannot test all these 40 strains or 50 strains that we have isolated from rhizosphere region, or hundreds of strains that we have isolated from the rhizosphere region in the field conditions, because if we go that route, then it will take a lot of time and a lot of resources, and we cannot complete all those experiments. If we wanted to test it in tomato, then it will take like 6 months, and also you see the resources. So in that case, we could use the molecular techniques to screen all these strains to see what or the mechanisms, or what are the genes that are present in those microorganisms, and based on that we can correlate with its antagonistic activity that have been observed under in vitro conditions. So then we can narrow down the number of microorganisms from hundreds or from thousands of microorganisms to the elite strains. It may be anything in number. So here this just I explained the exploration of PGBR strains and testing of all individual strains would be a tedious process considering real crop cultivation processes. So we could say these genes that are responsible for growth promotion are genes that are responsible for the antagonistic activity, and those could be used as the markers as being used in case of the selection of plants. In case of plant breeding, we know that they use the genetic markers to locate or to select or tag the resistant germplasms or the elite germplasms. And similarly here, we could also look, or we could also use the same methodology to look for the best or elite strains. So in this case, the handy strategy, it would be screening hundreds of microorganisms based on the functional genes already identified with an aid of molecular techniques. And here, as I told you, if you have like 100 strains, then you could screen all those strains, and you can look for all those genes, and you can pick up those strains without missing any elite strain. That is one advantage would be here. Then also the other thing is, once you have identified these strains with the different mechanisms, it is not possible for all microorganisms to possess all properties, and some may be very good in promoting growth, but at the same time it may not be effective in suppressing a pathogen. But whereas in other strain, it may be very good in suppressing a pathogen, but at the same time it may not be promoting plant growth. But there may be a different genome genes that are present in that particular in a particular organisms. So in that case, what you're supposed to do is, once we know that which genes or which mechanisms exist in a particular microorganism, then we will be able to combine these, and we will be able to make a microbial consortia with multiple modes of action to enhance the plant health and improve plant disease control. Also, these microorganisms that could serve as the source for identifying different genes, and those genes could be used to build the superior biofertilizer or biopesticide strains via genetic engineering, or else the simple method of enriching those genes to produce more and more, and we could actually harness the effect, positive effect of those organisms. And not only the genetic engineering of microorganisms, but in case of the genetic engineering of plants, we will be able to use these genes. If you see the BT plant, the biotech plant, and it has come from the Bacillus thuringiensis, the microorganisms which has the Bt toxin, and the gene which encodes the Bt toxin has been mobilized into plants, and it's being used as a biotech crop. So those microorganisms, once we identify and once we have a particular gene characterized, and that could be a very good source to mobilize into plants to produce the genetically modified plants. Now, as I told you, we have different mechanisms, and here growth promoting genes, and the most studied gene here is indole acetic acid, that is actually IAA. And here, similar to plants, the bacteria, it has also the pathways for synthesis of this plant hormone. And here, the bacteria such as Azospirillum and Pseudomonas, they have specific gene like aminotransferase as well as decarboxylase, and this aminotransferase and decarboxylase genes that can facilitate the higher production of indole 3 acetic acid. And also these bacteria, they have the potential to secrete these phytohormones, and this could be very well useful in plant growth. And in this regard, there are genes, that is indole acetic acid decarboxylase genes, have been sequenced. And with respect to the genes, again, we have to keep it in mind, and some of the genes may be a single gene and some of them may be a cluster of genes. So when we have that a cluster of genes, at our initial screening, we have to look for which is the critical one to be involved in that property. Here, if you see this IPTC gene, it is actually considered as the critical gene or critical enzyme to produce IAA when compared to the transferase gene. So in that case, we have to look the specific genes and the key genes and key enzymes and the primers, and that has to be used to screen those microorganisms in such a way we will not miss the ones which may have those genes. But if you are using the primers or the ones which is not appropriate to tag to identify those critical genes, then we may miss those allied strains as well. And here in our study also, we have done a lot of growth promoting studies in seedlings or in short-term crops. And here we have observed the growth promotion when it compares to some of the other microorganisms and the untreated control. And here, if you see these strains, and here and this one are showing very good growth promotion, but again, we are not sure whether it is IAA or some other unknown metabolites are produced. And even when we tried with this IPDC gene and it doesn't work, so it indicates that it may have some other mechanisms other than that. And in that case, we are actually in the process of extracting the hormones and I mean the metabolites, and to study what it could be, and probably I will explain that later part of my presentation. Then with respect to Acetobacter, also we have isolated and we have studied its growth promoting activities. And when we try to see whether it has this nitrogen fixation genes, here if you see, I told you that the genes, it could be a single or it could be a cluster or polyketide synthesis genes. If you see here, there are many genes, but of this H NIF, nitrogen fixing F, this is a critical one to start the catalytic process. In this one, if we have identified uh, the, uh, uh, the microorganisms which could potentially promote growth, and if you are able to identify this gene, then it says that it has some property, and it could also be kept further, and it could be used for further testing and analysis. The next important one that we look to select or screen the microorganisms is growth promotion through stress mitigation. And here, whenever the plants are exposed to stress conditions, the plants, they are known to synthesize stress ethylene through the production of S-adenosyl methionine through aminocyclopropane carboxylic acid. Here, what happens? The plant, whenever it is exposed to stress, and this can produce stress ethylene, and this stress ethylene could cause senescence as well as growth retardation. But the plant growth promoting microorganisms such as rhizobacteria, they are known to possess unique enzyme called ACC deaminase, and this enzyme can act on the ACC that are synthesized in the plant, and it can actually convert this to alpha ketobutyrate and ammonia, and it could prevent the accumulation of stress ethylene. Thereby, the biotic stress or abiotic stress could be managed. And here, this is my work done a few years ago. And here, what we have done, we have identified the strains which possess that ACC deaminase activity using the enzymatic as well as the PCR assay. Then after that, we tested that strain with the strains that do not possess ACC deaminase activity. And when we have tested it in salt stress conditions and the conditions without salts and salt stress, we have seen that the bacterium which possesses this ACC deaminase activity could enhance and could perform very well and could enhance plant growth under salt stress conditions as well. But whereas in case of the other microorganisms which do not have the property of ACC deaminase, but also they have other growth promoting properties and the antibiotic genes, but however, it do not have that particular gene, and it indicates that this ACC deaminase could be the reason for promoting growth under stress conditions. So with respect to growth promoting genes, these two are the major ones. And the next mechanism mostly studied in case of biocontrol is antibiotic genes. And here, antibiotic or antibiosis is a very important mechanism to suppress the growth of the pathogens. The reason is pathogens, they are very virulent, and once they attack the plants, then we should have some mechanism to suppress the activity of those microorganisms. In this regard, there are so many antibiotic genes have been studied from Pseudomonas fluorescens or Chlororhapis and Pseudomonas putida. Here, there are primers that has been available to identify those antibiotics such as diacetyl fluoroglucinol and the phenazine carboxylic acid. And so these are all some of the yearly primers or studies or preliminary studies can be performed on the number of strains to see which strain possesses these properties so that it can be taken forward for further processing as well as testing. And here, if you see from the whole genome sequencing of this Pseudomonas fluorescens, it was done in 2005 by researchers, and they have indicated that this low molecular weight antimicrobial compounds, which could arrest the growth of the pathogens, and they are present in the genome of the Pseudomonas fluorescens bacteria. And here, if you see this ophide and hydrogen cyanide and pyrolutrine and pyrochelin and pyrrole nitrine and pyrovidin, and these antibiotic clustering genes, it contribute about 6% of the whole genome. So this indicates that this could be a very good candidate gene for screening of the bacteria if you are working on the plant growth promoting microorganisms in the perspective of a disease control agent. Then here also we have, as I told you, we have isolated several Bacillus strains, and in case of Bacillus, the lipopeptide genes have been studied, and those lipopeptide genes have antifungal, microbial, and antibacterial and anti-oomyces, and some of them have the anti-nematode and antivirus properties as well. And here what we have done, we have used different primers to detect the genes that are present in our strains, like iturin and bacillomycin and fungicin and bacillicin and surfactant, using different primers as well as the different PCR conditions. And we have seen out of these 47 strains which we have tested, all these Bacillus amylolipoacin strains, and most of them, they possess this iturin C and D, whereas in case of bacillomycin and bacillicin, if you see, some of them they do not have. And here, this blue color, it indicates all Bacillus amylolipoacin strains, whereas this green, it is Bacillus megaterium, and this purple is Pumilus, and here this orange, it is actually Bacillus cereus. I told you that we had more diversity of Bacillus strains other than these four groups, but we haven't seen any of the lipopeptide genes amplified in those strains. So this indicates that if you see these strains of Bacillus amylolipoacins, and they possess all these lipopeptide genes that we have tested in those strains. So this indicates that this has some antagonistic potential. And interestingly, we have sequenced all these 47 strains of the 16S or RNA gene. And when we have used this data to analyze the gene, and we have seen that the antibiotic producers, this is not based on the lipopeptide gene, but based on the 16S or RNA gene. And we were able to see that these antibiotic producers, they all grouped in, all clustered in one group, whereas the group 2 and the group 1, and we haven't seen any of these lipopeptide genes present in those microorganisms. So this indicates that the 16S or RNA gene could also be very well used to study the diversity of the microorganisms. And in addition to that, it has very well clustered the antibiotic producers. And when we tested these antibiotic producers against vegetable pathogens, such as Fusarium wilt in tomato and Cercospora lispot in lettuce, which is caused by Cercospora lacticae, and bacterial wilt in tomato, which is caused by Ralstonia solanacearum, then anthracnose in hot peppers, which is caused by Colectotrichum capsicum. And here you could see a clear zone as well as high antagonistic activity against these strains. But when we tried the non-producers, and we haven't seen any of the inhibition activity, and it indicates clearly that these lipopeptide genes, they have some potential to suppress the growth of the pathogens, and we are on the right track to identify the elite strains. Then we have tried the strains which possess all these genes against the leaf spot disease in lettuce, and we found that the lipopeptide producing PGPR strains were able to suppress the growth in plants under greenhouse conditions, and it promotes growth as well. Also, we have conducted a field trial, intensive field trial, and that also it has been noted that it could suppress the bacterial leaf spot as well as Cercospora leaf spot disease. Then what we have done is here, we wanted to extract those lipopeptides and wanted to study if those extracts have any other unique compounds in it, and we have extracted these lipopeptides, and we have done the fractions and using column chromatography. And when we have done those fractions, and one of the fractions has showed specific activity against a fungi, and one of them has showed a specific activity against a bacteria, and this indicates that that has a specific antifungal as well as antibacterial activity. So we the next process is actually using multi-dose mass spectroscopy as well as the NMR to identify those unique compounds. So in that way, suppose if we miss any of those compounds, then using this column chromatography as well as the mass spectroscopy and the NMR using the chemistry or the chemistry based techniques, we will be able to identify those organisms. Those lipopeptides or those antifungal or antibacterial compounds. When we tested those fractions, and you could see the lytic activity of those fractions on the spores of fungi that cause disease in lettuce. So if you see the lipopeptide genes that has been widely reported in Bacillus strains, and as said, the whole genome sequencing cost has come down, and a lot of sequencing of those identified strains or LA strains have been done with workers, researchers, and here the outer circle, it is Bacillus ameloleukophaciens subspecies and plantarum, and the second one, the Bacillus ameloleukophaciens subspecies ameloleukophaciens, and the third circle Bacillus subtilis, and fourth one Bacillus licheniformis. And here, if you see some of the lipopeptides, they are unique to those bacteria. For instance, if you see this Bacillus ameloleukophaciens, this is also a particular strain, and here you could see this plantazolin is a lipopeptide present in this unique, though all of them are belonging to the same species, Bacillus ameloleukophaciens, but still you could see the unique presence of those lipopeptides in those microorganisms. And here, if you see this one fungicin and bacillin and bacillicin and surfactin and this bacillibactin, and they're all present in all of these subtilis and ameloleukophaciens. But whereas the other colors that are indicated in blue and red and also in green, and they are unique to the particular Bacillus organisms. Here they have done an extensive work, this particular researchers, and it has been reported from this particular strain. As I told you, there are lipopeptides, and if you see, they have shown virus, antiviral, and antifungal, and also antibacterial activity, and also some of them have activity against anthrax and nematodes. So this indicates that these lipopeptides could be a candidate genes for screening and for preliminary screening and identification of the antagonistic strains. Here, as just I mentioned, that using multi-dose, we'll be able to identify the newer molecules, and also we'll be able to purify, and we could test those fractions and whether they are having a particular activity or broad spectrum activity. Then the last mechanism, which also we could think of exploiting to screen the microorganisms, is the PGPR mediated disease resistance in plants. And here what happens when the plant growth microorganisms associated with the plant, and they are known to induce the defense mechanisms which are in a sleeping mode in plants. So the plants and microbes, they communicate to each other, and they have the specific interactions like the PAMs, pathogen-associated molecular patterns. Here, the microbes, the MAMs, microbes associated molecular patterns, and they also talk to each other, and they trigger those defense mechanisms. And here, if you see the defense mechanisms and pathogenesis-related proteins, and they could be assessed using quantitative PCR to study the expression of genes, and we have also studied the expression of defense genes in capsicum treated with biocontrol agent and inoculated with phytophthora. Apart from qPCR, we could also use gel electrophoresis, maybe one-dimensional gel electrophoresis or two-dimensional gel electrophoresis to identify the differential expression of proteins in plants, so that we will be able to tag those proteins and characterize those proteins using the same multidoph, and we could identify that's a kind of a reverse technique. Using that, then again we could identify the gene or the enzyme which is responsible for the resistance. And here, if you review the literature of plant defense mediated through rhizobacteria, mostly it is jasmonic acid and ethylene mediated resistance. Here, the NPR1 gene is responsible for inducing these defense responses in plants, such as helicase enzyme or chitinase or defensin or vegetative storage proteins or lipoxygenase or phenylalanine ammonia lyase. Or many genes have been identified, as well as these transcription factors activated. And here, so based on these differential expression of proteins, we will be able to see whether the
Dr. Sarah, I think you're muted.
Okay, are you hearing me?
Yes, that's better now.
Okay, thank you. So was it so long?
Just a few seconds, but it was noticeably just chipped out.
Okay, thank you, Dr. Chan. So using these defense enzymes, we could identify the genes like chitinase or peroxidase. But now the 2D gel electrophoresis is the one which actually separates the samples based on the isoelectric focusing point and based on its molecular weight. In case of two-dimensional gel electrophoresis, and there is again an advancement, and using DIGE, differential expression of gel, and here you could use 3 samples or in a gel when compared to this 2D gel electrophoresis. And here the first step is you have to prepare the samples, protein extracts. Then it has to be used, and it is separated based on isoelectric focusing. Then it is actually separated based on its molecular weight and size. Here we have used the same in hot pepper plants inoculated with phytophthora and treated with biocontrol. And here, if you see, this one is phytophthora inoculated plants, and this one is biocontrol plus phytophthora, and this one is just biocontrol inoculated plants. And here when we sample the extracts, and when we have done this analysis, and here in this gel differential gel expression, and here we found that there are about 51 spots differentially expressed, and of these 51 spots, and several of them upregulated and several of them downregulated, and we picked up some interesting 10 spots based on its increase upregulation as well as downregulation, to see what's happening there. And we noticed that the biocontrol treated one, and it could enhance the production of mannose-rich isoform, and this is actually associated with a defense mechanism as well as palatine like isoform, and this is also reported in potato to offer resistance against pathogens. And here we have seen some of the defense responses here in this differential expression studies, but it would be good to characterize all the interesting spots to see what happens, and this has been done using multi-dove analysis. Apart from studying these mechanisms, we know that once you have identified a strain using a lot of these screening studies and also conducting the trials, then also you could do the whole genome sequencing of a particular organism. As I said, the cost has really come down after the era of next generation sequencing, such as using Illumina or ion torrent or FIRO sequencing. Now the longer fragment sequences has also come, like Nanopore and Pacific biosystems, and those could also be used. And here, based on that, several microorganisms that have been identified as a plant growth promoting microorganisms or identified as a biocontrol strains have been sequenced, and a lot of mechanisms or the genes that has been unraveled from those sequencing. So with this, I summarize my presentation that plant growth promoting microorganisms are suitable, are very well could be very well used for plant disease control. And molecular techniques could also be used to tag the allied strains or to screen hundreds and thousands of strains based on the known mechanisms such as growth promotion, antibiosis, and induction of defense mechanisms. And the molecular techniques and whole genome sequencing could be used to unravel the mechanisms involved in plant health due to plant growth promoting microorganisms. So thanks very much for your time. I know that I'm going beyond the time. If you have any questions, then I'm ready to take the questions. Thanks so very much.
Dr. Saravanakumar, I have a quick question.
Yes,
the biocontrol agents that you use, especially the soil agents, how effective are they? Or does the efficacy fade out after some time? Or what is the interaction of those organisms with the natural microbial community? Or these factors that are taken into account based on your experience? What's your experience on those?
Okay, so with respect to that, what happened? The first question is efficacy. With respect to efficacy, the bacterial organism that we have introduced have declined over the period of time. So we have to apply at a particular intervals, and that we have designed experiment at different intervals, like week interval and 15 days interval and 21 days interval, to see what happens there. And in that case, we have seen that the efficacy has varied between these interval applications. And once you have it at 15 days interval, and that could has provided relatively high efficacy compared to the 21 days application. So it indicates that the community that we apply, or the microorganism, the microorganism which we apply is also declined. So for that, definitely we have to study some of the enhancing factors or else the augmentation factors such as the media or else the formulation types. So that's what actually we are also doing it, the different types of formulation so as to enhance its viability and competition in the environment to enhance its efficacy. And you ask about what happens to the community of other microbes. And unfortunately, we haven't done that part as well. And having assumed that we have isolated the microorganisms from the rhizosphere or from the nature, and we thought that it will not have much impact. It's an assumption. I'm not certain. Yeah.
One more additional question there. So when you apply it, is it a localized application, or is there a way you need to apply it, or is there a difference when you're doing the greenhouse experiment and in-field experiment in terms of how these organisms behave?
So with respect to application, it is the same way, because we formulate and we apply in greenhouse, and the same way of application is also going into the field.
Okay.
Yeah.
Thank you.
Yeah, you're welcome. So any questions? I see now we go into the discussion. I think we have our speakers as well. Davide be here. Yes, he is here. And then we have Dr. Shanmugam. I see a question for Davide. Davide, shall I ask the question? Okay, first I will ask the question, then we will go with Chinna, Dr. Chinna Durai. First, the question is from Farah. Is there a LAMP assay for the detection of the lethal yellowing pathogen in coconuts? If yes, how can this technology be made available to field researchers?
Well, I'm not aware about this disease. I know it's caused by phytoplasma, and there are other LAMP assays for other phytoplasmas. So I believe it could be developed. It's mainly a question to design 6 primers on a particular region. So I would say that it's possible to develop, but I'm not aware about this.
Okay, so in case of phytoplasma, I have a little experience in working with this coconut phytoplasma, and there are qPCR assays have been very well developed for the detection of this phytoplasma, and also the issue is probably we need to isolate, and we have to fully characterize the strains that are present in the region, and based on that we will be able to develop the primers based on the sequences, and then the LAMP and other assays could easily be made available to the field researchers in the region, because the strain variations is also a key issue with respect to minor organisms such as viruses and phytoplasma, the strainal variations. So of course the LAMP, as you said, it has 6 primers, and so that the specificity will be enhanced. But qPCR assays are there, and one of the LACNO in the region could be, I say, since I'm here and I could represent the region. So we do not have much sequencing of those organisms or reporting of those organisms at that level, so that we may not have the information to tag the primers or design the primers to make use of the field assay and other things.
Okay.
David, if you, yeah.
Anyway, I say maybe this could be a good source for collaboration, because yes, we have quite good experience now in the design, but yes, we should have some sequence to start. So I don't know if 16S of this phytoplasma is characterized, or maybe.
No, primers are there, but we have to tag and we have to sequence it fully, and we have to see is there any variation. I know it says in India and also in some of the countries it is present, and here also in it is restricted in some of the islands. So it would be good to do that, and also it is transmitted by insect hopper. So we have to see that as well. So I see a question from, but just I wanted to inform the house as well. We had a collaborative proposal to detect the quarantine pathogens that are affecting nuts and fruits in the region with Davide, and we submitted to you. But after it passed to two different stages, and finally they said, though the project was very good and is effective, but they weren't able to find the fund for those research. So it's very unfortunate, but I'm sure that we'll have some sort of collaboration going forward. Thank you, Davide. So I request Chinna to unmute his mic, and please you ask your question.
Hi. First, I want to wish all the four presenters a very good presentation and very informative. And I congratulate all the four for giving these wonderful presentations. So I also have the same question to Dr. Davide on loop-mediated amplification assay, LAMP assay. So I know the primer designing is quite difficult in LAMP assay for the detection of pathogens. But nowadays I'm reading papers with multiplex LAMP assays. So I want to know how it is difficult to design primers targeting multiple number of genes or multiple number of pathogens in the same amplification assay. Is there any specific tools or softwares available for designing primers specifically for the multiplex assay in LAMP?
Yeah, I would say that there are some available tools you can find at Aiken website, or even there are some other available tools, but from our experience, at the end, we have almost always designed them manually. So we use the tool and then we also try manually, and they work better. So I would say that it's a try experience. So, but what I would say that for multiplex, I believe it's quite complicated because you have to design several primers. So either you just select to change maybe the reverse primer or the forward primer, and you have maybe two species which are one close to the other. So then you can use the LAMP for two different species. Otherwise, it's a bit complicated because 6 primers will create dimers, or maybe they will interact one with the other. So the risk is not to amplify the real target of your LAMP.
Okay, but is there any specific tools for designing primers for the multiplex assay?
No, not for the multiplex specific, only for one species.
Okay.
Yeah.
Okay, thank you.
Thank you, Chinna. I see a couple of questions in chat box. The strains with biocontrollability you obtained, were they able to compete with the indigenous microbes in the soil on the field and still perform its biocontrollability, or was the study based on greenhouse experiment? Most often strains that are successful in the greenhouse often fail to perform on the field, and how can this be prevented? Okay, so this is a very interesting question because we also face, and here the indigenous microbes, right? And in this case, if you see the microorganisms, it has been already isolated from the microorganism. So it has that same niche, and only it has less competition with those indigenous microorganisms, because the reason is when you isolate those microorganisms, and you cannot miss the microorganisms which are high in population. So and also in the rhizosphere region, and it has less competition from the other microorganisms. Then you said that the experiments have been conducted only in greenhouse or in field trials, but we have conducted in greenhouse with the inoculation of the pathogens. And in addition to that, we have also tried in field conditions, and we have conducted the field trials. And based on my experience in this field for about 15 years, and I also say that the consistency of the performance of these biocontrol agents, it may not be like chemicals like the 100%, but it has to be applied from the beginning of the plant, like since it has the growth promoting activity, and it has to be applied as a soil application, as well as the foliar spray, and starting from planting into the field. So that way you can enhance those microorganisms, and you can enhance the health and the strength of the plant, so that the inconsistency can be prevented. But once we are going to apply the micro or this biocontrol agents after seeing the disease, then there may not be 100% success. So it's a pre-application is always a benefited in improving plant health and for better disease control. And Davide also has a lot of experience in biological control, and if he has something to contribute, I always invite, and also I invite Dr. Shankar if he has something to contribute to this question. So if I take the next question is from Sherian. How easy is to produce a stable commercial product from these strains, PGPR strains, that farmers can store, utilize for themselves? Okay, so to answer this question, there are different formulations and different carrier materials and inert materials, and also the adhesives, which should not affect the viability of the micro or the biocontrol agents. So those has to be studied, and we have done it. And so using different inert materials and carrier materials, it has been tested so that the shelf life of those microorganisms can be in the formulation from about 6 months to 1 year. So there are different type of formulations, like solid, liquid, or the semi-liquid formulations, and the slow release formulations. So we actually successful in one of the methods, and but we have to try, and we are successful in one of the methods. And so it could help the farmers to keep the shelf life of the biocontrol agents or the PGPR. Yeah. I don't know if I answered this question. So if you have more questions, you please feel free to unmute your mic and talk to the speakers and ask the question. Though we have here in the region, when we talk, one of the issues is actually everything, the consumables and everything, because all are small island developing states, and we do not have the consumables as well as the chemicals readily available here, like what we get in Europe or in US, and also the instruments. So everything, it has to be imported, and even a chemical. And during this period, it's very difficult to get the shipment of a product to do research. So really, there are constraints, but within the constraints, we are doing our best to do some of these techniques to accomplish the research. So
If there are no questions, I know that I told that the discussion will start 2 o'clock and it will close by 2:30, but it's now 2:45. If there are no questions, I sincerely thank every one of you for your kind patience and joining us on time and staying with us till now, till the last minute. I thank everyone. Um, I see a last question from Tina. So just, is PGPR more effective than IDM practices? So this integrated disease management practices is always welcome. And here this PGPR is also one of the component in integrated disease management strategy. So in that case, I cannot say that PGPR is standalone practice to do magic. So definitely it has one of the components in IDM strategies. And of course, all those IDM strategies such as the crop cultivation practices and the other practices that will always help to keep these plant growth promoting organisms. As asked by Shankar, the favorable conditions should be there, and otherwise then there will be inconsistency and there will not be a success to the biological control. So in that regard, definitely they are all complement strategies and we cannot separate them from each other. So hope I answered.
Yes, thank you, Sarah.
Thank you, China.
Sarah,
yes, Davide, please, yeah.
Maybe I add just one
sure
idea which is developing now in Europe, because you know, we have integrated disease management on one side, and then we have organic farming on the other side, because conventional agriculture is banned in Europe since 2014. So there are only these two types. But nowadays there is a third way in the middle, which is gaining more and more interest, which is the zero residue production. In the zero residue production, you still can use some chemicals, but at the time of harvest or when the product is on the table of the consumer, then there should not be any more pesticide residues. And it's becoming also an interesting way also for labeling and commercializing products, because it's not really elimination of pesticides, chemical, but it is a very high reduction of their use.
Thank you, Davide. Yeah, I understand that, because Europe, I always say whenever I do presentations, always stringent in the regulations, as well as they're very concerned about these highly hazardous pesticides. And most of the, even I heard that mancozeb is the one recently identified as the HHPs, and there is some concern going on with that. So really considering those chemical residues, and we definitely need to think of the alternatives. And hope also it's not only that, we also have some crops identified as the export crops, like hot pepper, for instance, if you see. And there is a Caribbean diaspora in Canada and UK and Florida, and they export a lot. And when it comes to pesticide residues, then again there would be an issue. So it's for the local consumption as well as for the for meeting the export regulations, we need to be reminded all the time. Thank you for that valuable point. So once again, if there is no question,
I just have one final question.
Yes, please.
I have this question to Dr. Shankar. I just want to know how they determine the depth of sampling based on the site in metagenomics. So how depth they are taking samples?
Okay, it depends. There are a lot of experiments on varied diversity based on the depth. It depends on what question you have. Like if you're more interested in the soil-plant relationship, and depending on the crop that you want to test that, you have to stick on to the A horizon, or the horizon of dominant root activity, which is close to 0 to 15 centimeter. That's the standard that we use for most of the cases. Because I work mostly on agricultural systems, so I tend to stick into that range.
Yeah, because the soil factors is also affecting the microbial communities, right? So it depends on what depth we are taking samples.
Yeah.
The proportion of communities may change. Am I right?
Oh yeah, it does. It does. That's why, like I said, if you are dealing with crop plants, where you are just looking at root activity in the air horizon, that's where
Yeah.
that's where now you want to look, because you're looking at how the nutrient is cycled through that area, so that how beneficial it's going to be for the plants. But for my PhD thesis that I did, when I did the sand dune clonal sequences, I went for 2-3 depths.
Okay.
0 to 15, 15 to 30, just to see how the overall soil development is happening in those dunes. Again, it depends on what question you're asking. Of course, yeah, there is this feedback. Soil properties will affect the microbes will affect the soil properties. It's both ways.
Yeah. Thank you.
Okay, thank you everyone. And before we close, I have an instruction. We posted the evaluation form in chat boxes, so the participants kindly go to that link and evaluate the webinar, because your feedback is always very important for us to have or to organize a webinar similar kind for the next time. So we always value that feedback, and thank you very much. And thank you very much to Davide. He's my very good friend, and Shankar as well, my classmate, my undergraduate batchmate. So I thank Prof. Ravindran as well. And thanks to everyone. Thank you very much. Stay safe. Season greetings, and I hope you will have holidays from next week. So enjoy your holidays, and happy 2020. We hope. All the best. Stay
Thank
safe.
you very much. Thank you very much for having me, and I really enjoyed it. Nice to have an acquaintance. I hope it will be carried forward in the future as well.
Thank you. Thank you.
Thank you.